Good day, this is Professor Will, CBMD PhD. Today, Sunday, December 9, 2018. It is 4:21:34 seconds Eastern Standard Time. Science News. The naked eye alone is not enough to ensure the accurate diagnosis of skin cancer, says experts. Date December 7, 2018. Source University of Birmingham. Summary. The visual inspection of a suspicious skin lesion using the naked eye alone is not enough to ensure the accurate diagnosis of skin cancer. A group of experts have concluded. The review is published today, December 6, in the Cochrane Library as part of a special collection of Cochrane systematic reviews bringing together a large body of research on the accuracy of tests used to diagnose skin cancer. The reviews summarize research evidence assessing the accuracy of different diagnostic tests to support clinical and policy-related decision-making in the diagnosis of all types of skin cancer. The visual nature of skin cancer means that it can be detected and treated in many different ways and by a number of different types of specialists. Therefore, the aim of these reviews is to provide the world's best evidence for how this endemic type of cancer should be identified and treated. We have found that careful consideration should be given of the technologies that could be used to make sure that skin cancers are not missed, at the same time ensuring that inappropriate referrals for specialist assessment and inappropriate excision of benign skin lesions are kept to a minimum. There are three main forms of skin cancer. Melanoma and cutaneous squamous cell carcinoma, CSCC, are high-risk skin cancers with the potential to spread and cause death. A basal cell carcinoma, BCC, rarely spreads, usually remaining localized with potential to infiltrate and damage surrounding tissue. Key findings of special collection were visual inspection using the naked eye alone is not good enough and melanomas may be missed. Smartphone applications used by people with concerns about new or changing moles or other skin lesions have a high chance of missing melanomas. When used by specialists, dermoscopy, a technique using a handheld device to zoom in on a mole and the underlying skin, is better at diagnosing melanoma than visual inspection alone, and may also help in the diagnosis of BCCs. Dermoscopy might also help GPs to correctly identify people with suspicious lesions who need to be seen by specialists. Dermoscopy is already widely used by dermatologists to diagnose melanoma, but its use in primary care has not been widely evaluated, therefore specific research is needed. Checklists to help interpret dermoscopy might improve the accuracy of diagnosis for practitioners with less expertise and training. Teledermatology, or remote specialist assessment of skin lesions using dermoscopic images and photographs, is likely to be a good way of helping GPs to decide which skin lesions need to be seen by a skin specialist, but future research needs to be better designed. Artificial intelligence techniques such as computer-assisted diagnosis (CAD) can identify more melanomas than doctors using dermoscopy images. However, CAD systems also produce far more false positive diagnosis than dermoscopy, and could lead to considerable increases in unnecessary surgery. Further research is needed on the use of specialist tests such as reflectance confocal microscopy, RCM, a non-invasive imaging technique which allows a clinician to do virtual biopsy of the skin and obtain diagnostic clues while minimizing unnecessary skin biopsies. RCM is not currently widely used in the UK, but the evidence suggests that RCM may be better than dermoscopy for the diagnosis of melanoma and lesions that are difficult to diagnose. Other tests such as using high-frequency ultrasound have some promise, particularly for the diagnosis of BCCs, but the evidence base is small and more work is needed. Apart from a few exceptions, I was surprised by how poor the overall study designs were, especially in terms of accuracy documenting where on the clinical pathway patients were tested. Although some useful conclusions have emerged, for example on the role of dermoscopy, the greatest value of the research is to serve as a yardstick for designing future studies evaluating skin cancer diagnosis techniques on patients who are typically seen in GP and specialist settings. The research team said that future studies evaluating diagnostic skin cancer tests should recruit patients with suspicious skin lesions at the point on the clinical pathway where the test under evaluation will be used in practice. Further research is also needed to evaluate whether checklists to assist diagnosis by visual inspection alone can improve accuracy and to identify how much accuracy varies according to the level of expertise of the clinician carrying out the assessment. Well-designed studies of dermoscopy in primary care are needed, and the best ways of delivering dermoscopy training needed to be identified. Today's is Professor Will C. BMD PhD. Today, Sunday, December 9, 2018. It is 4:42:21 seconds Eastern Standard Time. Potential scene for tailoring treatment for acute myeloid leukemia. Rapid drug response screening for leukemia stem cells offers clues to relapse and to improving patient-specific therapies. Date, December 7, 2018. Source, University of Washington Health Sciences, UW Medicine. Summary. Rapid screening of leukemia cells for drug susceptibility and resistance are bringing scientists close to patient-tailored treatment for acute myeloid leukemia. Research on the differing drugs response patterns of leukemia stem cells and blasts may show why some attempts to treat are not successful and why some patients relapse. Research on the drug response of leukemia stem cells may reveal why some attempts to treat are not successful or why initially promising treatment results are not sustained. AML is a serious disorder of certain blood-forming cells. In this disease, certain early precursor cells in the bone marrow that usually develop into white blood cells don't mature properly. They remain frozen as primitive cells called blasts, unable to further differentiate and mature. These can accumulate and cause low blood counts that reduce the ability to fight infections, and low platelet counts that cause risk of life-threatening hemorrhage. Leukemia stem cells, the progenitors for the immature cancerous blood cells, propagate AML, and also play a role in the cancer returning after treatment. Cancer researchers are interested in how genes are expressed in the cell population, because this data may hold clues to resistance to standard therapies and answers to why some patients relapse. A study presented at the 60th Annual Meeting of the American Society of Hematology in San Diego looked at the drug response patterns of stem cells and blast cells taken from individual patients diagnosed with acute myeloid leukemia. The information was gathered through the high-throughput screening, a state-of-the-art method for quickly evaluating and testing many samples. The researchers found that leukemia stem cells and blast cells diverged in their drug susceptibility patterns, and also that these patterns differed from patient to patient. For example, blast cells S responded in the test to the drugs most commonly used to treat patients, but none were effective against leukemia stem cells. The researchers did find 12 drugs from 8 classes that seemed to preferentially target leukemia stem cells compared to blast cells. Many of them are not often used in patients with this type of cancer. The multidisciplinary team on the project included stem cell biologists, hematologists, medical oncologists, pathologists, computer scientists, drug developers, and others. In the laboratory study, the researchers compared the drug sensitivity of blast cell and stem cell populations taken from the same 6 patients. In doing so, they tested a custom panel of drugs, targeted agents, and drug combinations on the cells, and did genetic analysis for 194 mutations. The panel included both FDA-approved and investigational drugs. The unique drug susceptibility patterns observed in leukemia stem cells and blast cells are leading the scientists to hope that patient-specific approaches could be developed against acute myeloid leukemia, with the goal of improving the outcomes for people with this form of blood cancer. Today, this is Professor Will C. BMD PhD. Today, Sunday, December 9, 2018. It is 4:58:14 seconds Eastern Standard Time. Science News. A code for reprogramming immune sentinels. Date, December 7, 2018. Source, Lund University. Summary. For the first time, a research team has successfully reprogrammed mouse and human skin cells into immune cells called dendritic cells. The process is quick and effective, representing a pioneering contribution for applying direct reprogramming from inducing immunity. Importantly, the finding opens up the possibility of developing novel dendritic cell-based immunotherapies against cancer. Our so-called dendritic cells function as the immune system sentinels. Their task is to scan our tissues for foreign particles, such as bacteria, viruses, or cancer cells, and to devour them. They subsequently break down the particles into smaller pieces, known as antigens, and present them on the surface to the immune system killer cells, T cells. In this way, the killer cells learn which infectious agents and cancer cells they are to search for and kill. Due to these key features, dendritic cells-based strategies have been tested to treat cancer patients. However, cancer can affect the dendritic cells in such a way that they get lost or become dysfunctional. We therefore need to find new ways of generating dendritic cells for every patient. Now, for the first time, a research team in Lund has succeeded in obtaining dendritic cells by a process called direct reprogramming. They have identified 3 essential proteins, PU-1, IRF8, and BATF3, that are required and sufficient to change the identity of most cells to make them become dendritic cells instead. They have also confirmed that the same protein cocktail reprograms human skin-derived cells to dendritic cells. This study is now published and highlighted in the cover of the Journal of Science Immunology. From a tissue section taken from the skin, we can cultivate millions of cells and reprogram them to dendritic cells in a process that takes only 9 days. Our study has shown that reprogrammed cells have the ability to effectively capture and present antigens to kill cells in the same way as natural dendritic cells. The researchers can now direct the induced dendritic cells towards a particular target by presenting the right antigen to them in the test tube before introducing the cells into the organism. This finding opens up future possibilities to develop new strategies for immunotherapy against solid tumors and blood cancers beyond the treatments currently available. This represents an excellent opportunity to merge the fields of cellular reprogramming and cancer immunotherapy. Generating dendritic cells employing direct cell reprogramming is very attractive from a therapeutic perspective. Our studies may be useful in the clinic for generation of patient-specific dendritic cells. Cancer immunotherapy employs the cellular components of a person's immune system to fight cancer, and it was awarded with the Nobel Prize in Physiology or Medicine this year. By using reprogrammed dendritic cells, the probability of rejection by the organisms is lower, as these cells can be generated from the skin of each individual patient. Tumors often undergo a number of mutations, developing into a heterogeneous entity, which makes it more difficult for the immune system to identify them as a threat. In a more creative perspective, we now want to explore the process of dendritic cell reprogramming to develop a cancer gene therapy. We are aiming at injecting the 3 reprogramming proteins straight into the tumor, forcing it to present their own tumor-specific antigens. This allows the activation of the killer cells against tumor cells and may lead to their elimination. We have named this concept Trojan DC, an analogy with Homer's Trojan horse. The great potential of this technology for cancer treatment lead us to start a new company together with London University for development of this concept into a product that hopefully will reach cancer patients one day. Additionally, our studies open up the possibility of reprogramming other dendritic cell subtypes, taking advantage of their distinct functional features, a better understanding of the mechanisms that determine the identity of immune sentinels, and of how to use this knowledge to reprogram other cell types into dendritic cells could make these patient-specific cells useful in the clinic. Good day, this is Professor Will, CBMD, PhD. Today is Sunday, December 9, 2018. It's 5:58 seconds Eastern Standard Time. Science News. Chemo brains caused by malfunction in 3 types of brain cell study finds. Date, December 6, 2018. Source, Stanford Medicine. Summary in a new study explaining the cellular mechanisms behind cognitive impairment from chemotherapy. Scientists have demonstrated that a widely used chemotherapy drug, methotrexate, caused a complex set of problems in 3 major cell types within the brain's white matter. The study also identifies a potential remedy. The study, which will be published online December 6 in Cell, also identifies a potential remedy. A drug now in clinical trials for other indications reverse symptoms of chemo brain, as the condition is known in the mouse model the researchers found. Chemo brain is becoming more common as cancer therapies increasingly allow patients to live many years beyond their diagnosis. There are 15.5 million cancer survivors alive today in the United States, a figure expected to reach 20 million by 2026, according to the National Cancer Institute. But cognitive side effects of cancer treatment can be debilitating and prolonged. Adults may be unable to return to work, and children often struggle in school. It is wonderful that they are alive, but their quality of life is really suffering. If we can do anything to improve that, there is a huge population that could benefit. Scientists have long known that drugs like methotrexate impair all the body's rapidly dividing cells, but how much drugs affect the function of brain cells has been poorly understood. Cognitive dysfunction after cancer therapy is a real and recognized syndrome. In addition to exiting symptomatic therapies, which many patients don't know about, we are now homing in on potential interventions to promote normalization of the disorders induced by cancer drugs. There is real hope that we can intervene, induce regeneration, and prevent damage in the brain. Chemo brain is especially severe in childhood cancer patients, and children have the most to gain from better remedies. Inside the white matter, in addition to neurons which transmit nerve impulses, the brain's white matter contains other cells that help the neurons function. The research focused on three types of those cells: oligodendrocytes, which produce and maintain myelin, the fatty insulating shelf around the nerve fibers; astrocytes, which link neurons to their blood supply, promote proper connections between neurons and maintain the neuron's environment; and microglia, immune cells that can engulf and destroy foreign invaders in the brain, as well as sculpt neural circuitry. Comparing post-mortem frontal lobe brain tissue from children who had and had not received chemotherapy, the researchers showed that there were far fewer oligodendrocyte lineage cells in the brain than the chemotherapy-treated children. To figure out what was happening to these cells, the researchers injected young mice with methotrexate at levels designed to replicate human exposures during cancer treatment. The mice received 3 doses at weekly intervals. 4 weeks later, the researchers compared the mice's brains to those of mice that had not received the drug. Methotrexate chemotherapy was found to damage the brain's populations of oligodendrocyte precursor cells. Normally, these cells can quickly divide to replace any that are lost, but after methotrexate was administered, this self-renewal process did not happen correctly. More precursor cells than normal were starting down the path of maturation to oligodendrocytes, but they were getting stuck in an intermediate immature state. The same problem was seen in mouse brains 6 months after methotrexate was administered. Transmission electron microscopy in the mouse brains after methotrexate administration revealed deficiencies in thickness of the myelin insulation around nerve fibers, similar to changes in brains of humans who have received chemotherapy. Mice exposed to methotrexate also exhibited behavioral problems after 4 weeks that were similar to humans with chemo brain, including motor impairment, slower movement of their forepaws, signs of anxiety, and an open field test used to assess how threatened the animal feels in an unsheltered environment, and impaired attention and short-term memory function, evidenced by the inability to discern between novel and familiar objects, a symptom that persisted for 6 months after methotrexate was given. The researchers injected oligodendrocyte precursor cells from healthy animals into the brain of animals that had received methotrexate to see if the cells' maturation problems were caused by some aspect of the brain environment after chemotherapy. The precursor cells still began maturing at higher than normal rates, but did not get stuck partway through the maturation process, indicating that the brain environment was partly responsible for the cell's abnormal maturation. Cochlea Activation. Further studies showed that microglia, the brain's immune cells, were persistently activated after methotrexate exposure for at least 6 months. The activated microglia caused problems for astrocytes, the cells that help neurons get nutrients and function properly. Administering a drug that selectively depleted microglia to mice that had been treated with methotrexate reversed many of the cognitive symptoms of chemo brain and reversed the abnormalities in maturation of oligodendrocyte precursor cells, activation of astrocytes, and myelin thickness. The biology of this disease really underscores how important intercellular crosstalk is. Every major neural cell type is affected in this pathophysiology. She suspects this type of complex dysfunction may also underlie other cognitive disorders. I think that is probably more the rule than the exception. More research is needed to understand exactly how the different cell types are signaling to each other, as well as when and how medications could be best deployed against chemo brain. If we understand the cellular and molecular mechanisms that contribute to cognitive dysfunction after cancer therapy, that will help us develop strategies for effective treatment. It is an exciting moment. Today's is Professor Will, CBMD, PhD. Today is Sunday, December 9, 2018 at 6:25, 33 seconds Eastern Standard Time. Science News, new ways to look at protein RNA networks. Date December 6, 2018. Source German Cancer Research Center. Summary for their vital task. All RNA molecules in our cells require proteins as binding partners. Scientists have developed the first method which they can analyze the composition of the entire RNA protein network of the cell. RNA molecules perform vital tasks in every cell. Messenger RNA, mRNA, helps to translate the genetic information stored in the DNA into proteins. However, many other RNA molecules exist which are not being translated into protein. In fact, only 5% of RNA in a human cell is an mRNA. For many of their functions, RNA molecules have to interact with proteins. Sometimes different types of RNA come together with specific proteins to form highly complex molecular machines. The best example being the ribosome, where protein synthesis takes place. A gigantic network of interacting RNAs and proteins is active in each of our cells, but we still know extremely little about the exact composition of this network. We want to understand which proteins bind to RNAs and how this differs between cell types. or in conditions when cells are stressed. We have now developed a method that enables us to investigate this for the first time. Until now, such analysis could only be carried out for one class of RNA, namely mRNAs. mRNAs are the templates that instruct protein sequence, protein interactions with all other so-called non-coding RNA types, some of which have only been known for a few years, could not be detected using the existing method. Non-coding RNAs by far outnumber mRNA molecules, and they fulfill various regulatory purposes. Research scientists now succeeded in developing a method termed xRNAx to analyze the interactions of all RNA class of cellular proteins. Using xRNAx, the scientists can also make quantitative statements. They can not only see what protein binds RNA, but also to what extent. In this way, they were able to observe how RNA binding changes when cells are exposed to a toxin. In addition, with the new method, the research team identified hundreds of proteins that previously were not known to bind RNA. With xRNAx, we are able to measure all interactions between protein and RNA, which is something nobody could measure before. Many protein-RNA interactions are suspected to be the underlying cause for diseases, including cancer, amyotrophic lateral sclerosis, or viral infections like HIV. Now we have a way to look at them. The dates is Professor Will C. BMD, PhD, today Sunday, December 9, 2018 at 6:41:13 seconds Eastern Standard Time. Science News. New imaging tools that trace key breast cancer enzymes may help guide therapies. Researchers use radio tracers to measure PAR, P-1, and glutamine in breast cancers. Date December 6, 2018. Source University of Pennsylvania School of Medicine. Summary: a set of emerging diagnostic tools may help identify breast cancer patients who are most likely to benefit from therapies that target important enzymes fueling a range of subtypes, including BCRA mutated and triple negative cancers. Today, some breast cancer patients with BRCA mutations and triple negative cancers are treated with poly-ADP ribose polymerase PARP inhibitors, which trap and destroy PARP-1, an important enzyme for DNA repair that cancer cells rely on for survival. Similarly, therapeutics that target glutaminyl, an enzyme feeding some triple-negative breast cancers, are also being developed. However, no method currently exists to measure these enzyme expression levels in patients. Having a biomarker to quantify those levels would help identify those most likely to benefit from targeted therapies in a non-invasive way. In the first study, abstract number 851093, researchers from Penn's Department of Radiology and the Abraham Cancer Center used PET imaging, a novel radioactive tracer known as 18F, FTT, to measure PARP-1 levels in 30 women with breast cancer with various subtypes before therapy, including surgery. The researchers correlated the FTT uptake in the breast cancer patients with untreated surgical specimens that had been immunostained for PARP-1 in the lab. They found that the PET imaging agent could effectively visualize and measure PARP-1 levels in all breast cancer tumors and metastases. Past studies from the same team of researchers have linked PARP-1 levels to targeted therapy resistance and shown FTT ability to quantify PARP expression levels in ovarian cancer. This study provides early validation of FTT as a quantitative method to measure PARP-1 expression in breast cancer. Importantly, it also shows that the level of PARP-1 expression varies considerably within a given breast cancer subtype, and that surprisingly any subtype can have a high PARP-1 expression. Many clinical trials with PARP-1 have focused solely on the triple-negative subtype. However, this new study suggests that some patients with ER-positive cancers, for example, may have high levels of PARP-1 and thus may benefit from this targeted therapy. What's more, the results provide anecdotal evidence that expression levels can vary considerably in BRCA mutation carriers. 2, which is important since BRCA status is often used as a criteria for selecting PARP inhibitor therapy. In the second study, abstract number 851760, another team of researchers from Penn Department of Radiology used a radioactive tracer as an imaging agent to measure tumor glutamine levels by PET imaging in the mouse models of breast cancer based on the knowledge that some triple-negative breast cancers rely on glutamine for survival and growth. While radioactive tracer PET has been approved by FDA for imaging prostate cancer, its abilities in other cancers are less known. This agent enters exist cells via glutamine transporters and undergoes minimal metabolism. Penn researchers unitized this imaging agent to observe what effect of glutamine inhibitor CB-839 would have on glutamine level in triple-negative breast cancer. Glutaminyl is considered a druggable target because it is a key enzyme of glutaminylysis, the pathway that cancer cell uses to metabolize glutamine in small molecule drugs. Assessing the tumor glutamine pool size will inform the pharmacological effect of glutaminyl inhibitors because the treatment induces an increase of tumor glutamine level. This imaging approach can potentially provide a precision medicine tool that can tell physicians early on which patients are responding and which are not. The researchers showed that radiotracer could effectively track the change of tumor glutamine level in response to glutaminyl inhibition. For example, cellular uptake in radiotracer was increased after CB839 was administered, consistent with a higher concentration of glutamine in the triple-negative breast cancer cells and a lower level of the enzyme activity. Compared to chemotherapy, which usually shrinks tumors quickly and can be accessed by measuring the tumor size, metabolic drugs such as glutaminyl inhibitors take longer to affect tumor size. Therefore, a non-invasive method that detects the early response to these drugs would be useful. These studies are important steps toward developing much-needed non-invasive imaging tests to help measure and predict inhibitor responses and guide treatments for women with a wide range of breast cancers. Today's is Professor Will, CBMD, PhD. Today, Sunday, December 9, 2018, is 7:07:44 seconds Eastern Standard Time. Science News. Newly identified T cells could play a role in cancer and other diseases. Date December 6, 2018. Source University of California, Los Angeles, Health Sciences. Summary. Researchers have identified a new type of T cell called phospholipids, reactive T cells that are able to recognize phospholipids, the molecules that help form cells' outer membranes. The scientists also discovered that phospholipids compete with glycolipids, another type of molecule that helps form cells' outer membranes in a way that prevents glycolipids from readily reaching the surface of the cell. Cell membranes are primarily made up of two types of lipids, phospholipids and glycolipids. Inside cells, these lipids bind to a molecule called CD1D that transports them to the surface. Once their phospholipids stimulate phospholipid reactive T cells, and glycolipids stimulate a different type of T cell called iNKTs. On their way to the cell surface, phospholipids more easily attach themselves to CD1D molecules, making it more difficult for glycolipids to attach to CD1D. Because of this, it is harder for glycolipids to make it to the surface of the cell. This means that iNKTs cannot be as easily stimulated by glycolipids. Scientists believe that iNKT cells are necessary because they appear to protect the cells against the progression of certain cancers and autoimmune diseases. However, iNKT cells are extremely active and can cause alcoholic hepatitis or other types of liver diseases if they are overstimulated. The phospholipids' ability to more easily bind the CD1D molecule than glycolipids keeps a balance between the two cell types and maintains homeostasis in the immune system. To identify the T cells that react with phospholipids, the scientists first utilized isoelectric focusing gel electrophoresis and crystallography to demonstrate the chemical and physical binding of phospholipids to CD1D. They then loaded CD1D tetramers, groupings of 4 CD1D molecules with phospholipids, and used flow cytometry that detected the T cells that recognized the tetramers. Scientists found that phospholipid reactive T cells are rare. They accounted for only 0.3% to 1.8% of lymphocytes in animals, liver, spleen, and bone marrow. They then used crystal structure flow cytometry and cell cultures to show that phospholipids readily occupied the CD1D groove and outcompeted glycolipids. Phospholipid reactive T cells need to be further studied so that scientists can understand their function in people with alcoholic hepatitis, dyslipidemia, cancer, and autoimmune diseases. If future research identifies changes in how lipid behavior influences these diseases, scientists might be able to stimulate or inhibit phospholipid reactive T cells to treat some of those conditions. Good day, this is Professor Will, CBMD, PhD. Today's Sunday, December 9th, 2018. It is 7:33:31 seconds Eastern Standard Time. Possible alterations in DNA involved in cancer. Date December 5th, 2018. Summary. The study shows the role of the protein PIF1 capable of undoing different structures in these molecules. These molecules contain the instructions that allow cells to function correctly, so that when there is an alteration that is not repaired properly, mutations can occur that can cause problems for the health of the body. To avoid these problems, it is necessary to keep the DNA molecule intact. So safeguarding the genetic material, however, the cell's own metabolism and especially its own use of this information means that there are habitable physical and chemical alterations that could compromise the information the DNA contains, cause mutations. In fact, the cell can suffer tens of mutations every day. As much as possible, avoid any loss of genetic information. Multiple mechanisms have evolved, and these are capable of fighting these alterations and repairing the DNA. If these mechanisms are not sufficiently efficient to repair all the alterations, mutations build up that are natural causes of cell aging, and occasionally can lead to the appearance of various pathologies, including cancer. Although much is known about the basic mechanisms of DNA repair that act when the molecule is in the state of normal configuration, still little is known about the additional factors required to repair the parts of the DNA that are configured atypically. In this article published in Cell Reports, we have discovered that protein PIF1, a protein with the ability to undergo various DNA structures, acts during the repair of DNA breaks, which can form a cross-linked structure known as G-quadruplets. These structures are produced when there are areas in the structure of the DNA that are rich in guanine, one of the 4 letters of the genetic alphabet, which can interact with each other. Our data suggests that the presence of these structures impede the work of the repair machinery unless they are undone by PIF1. For this to happen, PIF1 needs to interact with the protein BRCA1, known for its importance to this process and whose mutations cause breast cancer. Identifying this new factor, which is associated with the repair of DNA breaks, can be the long-term bring about a better understanding of the process that caused the accumulation of mutations associated with the appearance of tumors. In addition, better understanding what is happening could open new therapeutic options. Some drugs that stabilize the G-quadruplets are included in the clinical studies for fighting cancer, so knowing what factors are going to interact with these DNA structures will allow doctors to better choose what type of patients would best benefit from these treatments.